So thanks very much for the kind introduction, Sarah. It's my pleasure to be here today. This is an unusual setting for me to speak at a bar. I used to come to this bar. I live right here next door to have some drinks and so forth, and first time to talk science here. Anyway, I have been at EMBL since 2003, and in parallel I had a lab at Stanford, and I had the privilege to really partake in two revolutions in science, I would say. The first one was through the development of the ability to sequence an entire genome. And the genome is the DNA material that you inherit from your parents that's found in every single one of your 10 trillion cells that are found in your human body. And it encodes a blueprint for life. And so that was first successfully done for organisms around the time when I started my PhD in 1997. And I think the second revolution is the revolution that I want to talk about today, which is the revolution of how genomics is transforming the way in which medicine will be done. And I think it's going to have an impact on all of society. It's going to change the way we go about maintaining our health status. It's going to change the way we go about treating diseases. And I'd like to start with this slide, which is actually just from an announcement that was made a few weeks ago by the President of the United States, with an incentive to go out and start to look at the genetic information from 1 million Americans, and to use that information in order to make informed decisions about treatment options and about methods to maintain health status. This is not unique in the world. There has been an effort called Genomics England, which has taken place in the UK, with the idea to sequence the genomes of 100,000 individuals and to integrate that with digital medical records that are maintained by the NHS. But the idea of these efforts is really to take into account as much information as we can gather from an individual, their genome, as well as other digital measurements in order to make more informed health choices. And if one summarizes kind of what the incentive is behind kind of a precision medicine or a genomic medicine that takes into account DNA sequences, that we really want to move away from this idea currently where we're using one treatment to kind of a one-size-fits-all approach, where we're applying one set of treatments to individuals with certain sets of diseases that we all lump together, because that's kind of how we've identified these treatments, to an approach where we take into account the fact that we're all unique. We all have a unique genome sequence, and we all have been exposed to unique environmental factors that really determine kind of how these genes in the genome are expressed and which ones are starting to mediate an onset to disease, etc. So with the idea that kind of we're all unique, and you can stratify this first group of individuals into more subsets and then apply more targeted, tailored therapies. So some people describe it as being predictive, because based on the unique genome sequence and the environmental exposure of an individual, we can do some assessments about risks for developing certain diseases. And then, of course, if we're predictive, we also want to be preventative and so act early and prevent diseases from occurring. Personalized for the reason that I already mentioned, because we're all unique, and it needs to be participatory. The idea is that we want to move away from treating disease when it takes its effect in an individual and actually prevent it from occurring. But that means that we have to start to analyze individuals that are still healthy in order to determine when things start to go wrong. I want to put up another quote that I heard just 2 weeks ago at the Personalized Medicine Conference in Silicon Valley. And this is from Greg Venter, one of the individuals who sequenced the human genome in a company effort. And he said, life is a DNA software system. Change the code, change the species. Change the code, and you can also affect kind of an outcome of health and disease. And this is maybe a controversial idea, that the genome kind of contains information that is predictive in terms of determining who we become. And I will come back to that and kind of challenge this notion. The human genome was first decoded in a massive effort that took 10 years by an international consortium of laboratories to complete. It took $3 billion to sequence the first human genome. and the completion date was around 2000. A major effort, and if you want to kind of put it in philosophical terms, I think it's also a unique event that happened on Earth. It was the first time a species actually managed to decode its own genome sequence. Today, this has really become easy. Sequencing human genomes today only costs $1,000, and we can do that on a single machine that a lot of advanced laboratories have in stock. And it's really routine, and so this has now been done for thousands of individuals. And when you look at the sequence, you can see differences in the sequence composition, and the hope is to attribute those to specific differences in the phenotypes, in the traits and so forth that individuals carry. If you would look at the sequence, you kind of have no idea what it means. It's difficult to read. It looks something like this. It's written in 4 different letters, and the order and arrangement of these letters is what encodes meaning. We know information for maybe 1% of the human genome, but most of the genome, even now, 10 years later, is still very much a mystery. If you wanted to print it out, it would actually take 50 meters of books in order to print out this entire sequence. So it's a vast, immense amount of information. But unfortunately, changes in just a few individual letters in the sequence can have very drastic consequences. And here's one example. Cystic fibrosis is a disease, one of the first ones that was genetically dissected. And this mutation here, where you have a deletion of just these 3 positions, CTT, leads to this disorder, which has a life expectancy of only about 37 years. So individual changes can be hidden in the code and can have drastic consequences. Not all changes in sequence have drastic consequences, and luckily, it's really the interaction of genetics and it's the interaction with environmental influences that determines the extent on phenotype. And this kind of comes back to that quote that I put up from Greg Venter, because it's not just the genome, but it's also the environment that determines whether that sequence is detrimental or whether that's actually beneficial. Because you can have one sequence detrimental in one condition, but that same sequence could be beneficial in another condition. And you can consider kind of environment, what you eat, that can determine the degree of fitness that an individual displays. And I want to tell you one personal story kind of that illustrates this concept of how genomes and sequence interacts with environment. And one of these examples is when you think about taking medications or drugs. Because some of us kind of respond well to certain medications, and we don't respond well to other medications, and this is a unique property. And so this is actually a personal analysis of my own DNA sequence that was carried out by a company that offered this as a free test. And what they look at are so-called enzymes. These are proteins in the body that are involved in the metabolism of drugs that we intake. And you need to kind of get, when you're thinking about treating an individual, you want to get the dosage right. If you take too much of any medication, it's likely going to be detrimental and have toxic effects. If you take too little of a medication, it's not going to be useful at all. So you kind of want to be in the green range here in order to target this. And so what they did is they looked at the genetic composition for 8 genes in my genome, and based on the DNA sequence inferred how active these enzymes are. And so for 4 enzymes, it looks like I have normal activity levels, so I can take the drugs like anybody else would take. But for 4 other enzymes, I have low or reduced activity level. And this came with a set of take-home messages. And one of the things the company told me is, avoid cigarette smoke, barbecues, and caffeine. Because these are all things that are metabolized by N-acetyltransferase 2, the first enzyme here. And so, I wouldn't be able to break these down as effectively as the average person is. In addition to that, I should alter drugs that are going via this particular transporter called the ABCD1 transporter, because I have reduced activity levels for that particular enzyme. So if I take drugs that need to go into cells based on this particular protein, then that won't be as effective, and I'm going to see more side effects of these drugs because they're going to stay around in the bloodstream rather than being metabolized inside cells. So those are things like actinomycin D, erythromycin, cortisol. In addition to this, unfortunately, it looks like I have no activity levels for glutathione S transferase. This is a molecule that breaks down reactive oxygen species, toxic molecules in cells. And so I should drink some green tea because green tea is actually an antioxidant. Now red wine is an antioxidant too, but that wasn't on the list. In addition to that, I should avoid drinking grapefruit juice because grapefruit juice is actually an inhibitor of the ABCB1 transporter. So on top of having genetically already inherited reduced levels of that enzyme, if I drink green tea grapefruit juice on top of that, then I'm going to just completely suppress any remaining activity of that particular transporter. So that's bad news too. Now I want to tell you about a story that takes genetic information more into account rather than predicting kind of these more lifestyle decisions that I showed you on the previous slide to actually have an impact on the treatment of human diseases. And this is a story that is really spearheaded by this family that I'm showing here. They're a family that have a child with an ultra-rare genetic disease. When it was first detected in the children, there was no idea what the defect in this child could possibly be. And the family, though, took a very active role in putting together teams of scientists to start to study this particular disease, to try to figure out the cause, and now to try to figure out a strategy of treatment. And in a lot of ways, they've kind of funded a lot of the research, not only in our lab, but in other labs in this area. So what is this? So this is a child that has a defect in terms of showing impaired motor control, global developmental delay. So the child is delayed in terms of its development, shows some mental retardation. It has seizures, and what is very unique, it doesn't seem to show any tear production. All of the other symptoms kind of resembled something like an energy deficiency in this individual. However, no tear production was something that we had never seen before. So whole genome sequencing was done. We sequenced the entire 50 meters of books for this individual and then searched for mutations in this individual. And mutations were identified in this particular gene called NGLI1 in these individuals. And the child has 2 defective copies of that particular enzyme, while the parents only have 1 defective copy. So the child has inherited both defective copies from the 2 parents. This started then a whole effort by the parents of not just this family, but there was another family that had a child with the same symptoms for which genetic analysis revealed mutations also in this gene, to kind of hunt and search for additional parents and families that have children with similar symptoms. So one family wrote a blog post that you can see here on the right side, and that has also helped actually in identifying additional patients with this disease. And for example, one family in India, the mother read the blog post and then realized that her child's symptoms were very similar to this, and they sequenced the NGLI1 gene and identified a mutation there. Today, there are 22 patients with mutations in this gene. There's a new patient coming about every 1 to 2 months or so. In addition, the other family launched a foundation in order to collect additional funding and to support research efforts in this realm. And a lot of kind of media sources have picked up on this. The Spiegel, for example, had a piece, Die Kinder ohne Tränen, the children without tears. The New Yorker had covered, CNN covered this, etc. And a number of scientific papers have now come out to report the identification of this genetic variant. I want to show you one short video that explains what NGLI1 is for a very general audience, and how what this mutation kind of means genetically, and how you find it.
What is NGLI1 deficiency? Each person has 23 pairs of chromosomes. Chromosomes carry genetic information in the form of genes. Your genome consists of roughly 20,000 genes. We all inherit 2 copies of most genes, 1 copy from our mother and 1 copy from our father. The differences from one person to the next come from the slight variations in these genes. Genes are truly amazing. They are the instructions for how to make the building blocks of your body. Imagine your genome as a giant cookbook. A single gene, such as NGLY1, would be a recipe in this book. Just like a chef uses a recipe to prepare an elaborate meal, cells in your body use your genes to make proteins. Proteins do much of the hard but critical work inside your cells. Some proteins give cells their shape and structure. Others help cells carry out essential functions for life, like digesting food or supplying oxygen. For many proteins to function properly, a sugar needs to be attached. Like most components of the cell, proteins are degraded and recycled. During this cycle, the sugar needs to be cleaved off. To perform this chop, you need the protein that is produced by the NGLY1 gene. If you can't make the NGLY1 protein properly, you can't perform the chop or remove the sugar, and the protein cannot be degraded. This results in protein accumulation, in essence, a logjam in the recycling process. When proteins accumulate, NGLY1 patients develop severe problems in their brain, liver, and muscle. To learn more about NGLY1 deficiency, living with the disease, and finding a cure, please visit gracewillsey.org.
So in order to kind of spearhead trying to identify the molecular functions of what that gene does, besides kind of what you've seen illustrated in this video, the family has assembled a team of scientists to start to work on that. We think that the enzyme is involved in other processes in cells that we don't really know about yet. And we think that that will be crucial to understand and to come up with a therapeutic strategy. And one way in which kind of genomics is helping this process is through the generation of lots of data. So previously, kind of medicine or the study of kind of human cells has been based mainly on cell lines that are standardly established in laboratories and have been our workhorses for many years. And we kind of collect only minimal sets of information on a particular patient in order to make a therapeutic decision. I think in future, this is going to be very different. We have now an ability to generate cells from every single individual, from every patient in principle that comes into the office. We can differentiate these lines into different types of cell lines that allow us to get a better understanding about what gene defects cause inside cellular systems, and we're going to collect millions of data points on individuals rather than relying on just kind of handfuls of measurements on patients to make much more informed decisions about treatment options and to come up with therapeutic strategies. And I want to show you a second aspect, I think, of this project that I think really is novel. And that's kind of the idea that if you want to tackle some of these rare diseases, you also have to identify people that are willing to work on it. You have to be able to fund it. Now, unfortunately, from a pharmaceutical company's perspective, the sample size is really small. You have 22 individuals currently that might benefit from this particular treatment, which makes it difficult for them to launch funding and resources into this project. So I think we're going to see a different way in which kind of patient groups are going to catalyze science, and that's through reaching out to the scientists directly, trying to collect their own funding in order to support this through foundations and other efforts. And I think rare diseases really is an area that shows a lot of promise in future, because through this sequencing we can actually identify the causative mutation. And so I just want to show you this short video, which has been put together also by this foundation.
It's an uphill battle having so few patients. You have to convince both researchers and pharmaceutical companies that this is worth their time and effort.
NGLY1 happens to be a very critical part of the cell's metabolism, very critical. And it's going to be linked undoubtedly to a lot of more common disorders as we figure out exactly what it's doing.
Rare is not an excuse.
Rare is not an excuse. Rare only means the future has not yet been invented.
Rare is not an excuse.
Rare helps me understand the common.
We face an uphill battle. This is not getting funding outside of private donations. Our greatest hope and mission for the foundation is that we will understand this disease and that we will cure it, and that these children will lead an independent life.
Rare is not an excuse.
希少ということは言い訳にはなりません。
Rare is not an excuse.
I think we will find a treatment for MBL1. We're supported by a great family that's behind it and an excellent team of scientists that are working on it.
It gives me a lot of hope that we'll be able to do something that will benefit these children.
Rare does not define my daughter.
Every day that we wait is a critical day in the development of these kids.
We are rare.
So a lot needs to be done still to come to an understanding here. In addition to using genomic information kind of to treat individuals that are diseased, I think genomics is going to change the way we're going to look at individuals that are healthy too, and I alluded to that. The idea is kind of to prevent diseases from arising in the first case. In the case of Grace Wilsey, unfortunately, the disease already manifested itself very early. And now we have to think about coming up with treatments or cures for this disease. But for other diseases that have more later age of onset, one could think about preventing the disease before it actually arises, and thereby save a lot of money that's currently expended in the healthcare sector. And I think that's by widening our information from the genome to other sets of information that can reflect on health. One is family history, for example, that we already use. If you have other individuals with a certain disease, then you take that into account. Analysis of which genes are expressed in an individual is important. Looking at the microbiome, turns out we have 10 times more bacterial cells in a human being than we have human cells. And these are impacting on health in interesting ways that's just now being unraveled. Metabolomic measurements, social networks, if you want to think about infectious diseases and you know, so it would matter who you hang out with depending on kind of which infections you might acquire. And so I've kind of just a few weeks ago decided to participate as a healthy subject in a study that starts to collect this information for healthy individuals, because the idea really is that we need to establish baselines for what constitutes a healthy pattern in these types of digital data in order to determine when an individual deviates from that pattern and proceeds to disease. So in addition to genome, we'll collect transcriptome, microbiome, metabolome data every 3 months from me over the course of probably the next 2 years to collect this information and integrate it with that of other healthy individuals and some diseased individuals. So I think kind of what we learned from this is that genes control your potential, but they don't control your destiny. And by changing your environment, you can change kind of the effect of a particular mutation in the genome, and you can actually, you might be able to even invert it so that that's beneficial. And there are a few cases like this that are known. And everybody can take control of their health through the analysis. We need people to participate. We need to collect this information for healthy individuals. And lastly, I want to finish kind of with this more futuristic view, is that ultimately we want to be able to measure these things in real time, periodically, and we need devices that are able to detect kind of these measurements and to integrate them. And you have digital devices on you all the time. The iPhone is one example. We can already use the iPhone to measure some parameters, like blood rate. We can measure sleep patterns and so forth. But the idea really to move this into the detection of more molecular data by carrying, for example, biosensors that one can integrate in the human body. And we have projects ongoing in the lab where we're trying to develop biosensors that could detect the expression of genes in the form of radio transmitters that might be present even within individual cells that can send signals to the outside that are self-powered and that you could register on digital devices in order to measure whether things are going wrong, whether you need to see a doctor, whether one needs to do further investigations in a particular space. And with that, I want to finish and thank you all for your attention. I'd be happy to take any questions.
Thanks a lot, Lars. Does Android work?
Android should work.
Yes. I'm an Android fan. Yes. All right, so Android works, everybody. Does anyone have a question before I ask mine?
Yes.
Yeah, I just have a question. So at the end of the presentation, you're talking about preventative sort of clinical trials or clinical trials that could be aimed for preventative treatment.
Yeah.
And what I always wonder is for like a neurodegenerative disorder that you could perhaps predict that they're going to get later on, what would be the time scale of such a clinical trial? And do you think that like these pharmaceutical companies would invest in such sort of clinical trials?
I think they're already, so we already see Roche and Novartis that are starting to incorporate genome sequence information into their clinical trials that they have ongoing now. Now their idea is to, more at the moment, is to stratify patients into different subgroups based on their genetic composition so that they could develop so-called companion diagnostics where you take your genetic information into account to make a decision on what I showed kind of on the first slide, which treatment option would be most appropriate for this individual. Because for them, obviously, it's of great interest because if they currently, let's say, have a drug that is effective in a subset of the individuals, but they don't know how to stratify them, they get an average performance that isn't very stellar. But if they're able to substratify a priori, then they can show, much stronger effects, and it will be more useful. So at the moment, the clinical trials will be more short-term, but the standard trials are already ongoing. I think in order to detect things like you're mentioning, longer-term development of disease, I think that's where we're relying on healthy individuals currently to participate. Because I think the bottom line and what we learned from these data is that there's no such thing actually as a healthy individual. We're all patients. And we all have defects in our genes. It's just that the environment of some individuals was such that the disease went to development, while in other individuals the environmental influences are not there or they're masked.
Any other questions?
Okay, I will go. So my question is that, since you have your genome sequence, and you know, that you have certain, how's it called? Yeah, for example, some drugs might do you more harm than the average person. Did you actually test it?
I didn't go and test any of the drugs to see if they have detrimental effects. No. I'm trying to take the recommendations into account. So when I'm faced with a choice between coffee or green tea,
okay,
unless I really need the caffeine in order to make it through the day, then I tend to choose the green tea.
Anyone else? Yep.
You sort of concentrated a bit on a quite rare genetic disease here. What are the major genetic diseases which are going to be coming through the research and cure some of the outcomes to impact large populations?
I think we're going to hear in one of the next talks, we're going to hear about the genomic applications, genetic applications towards cancer and oncology. That's certainly a very active area in which a lot of kind of these companion diagnostics have been developed that are tailored to particular genetic defects in the tumor. So that for sure. Mendelian diseases provides an opportunity for even kind of smaller research teams to make a major contribution. Because some of these complex diseases, cancer, for example, is made up of many, many different types of cancers. And we first need to probably be able to stratify these, and if we can stratify, we're going to have better chances. These rare diseases are already stratified. It's a single gene defect in these cells that is causing these defects. Currently, there's an estimated 7,000 Mendelian diseases around. We know the genetic basis of about half of them so far. But the prediction is that through the sequencing, you can see kind of a rampant increase in discovery of these genes, that we'll be able to figure out the genetic defects of these, the genetic causes for a large proportion of them. And now then the goal is kind of to use these more genomic approaches of transcriptome and other measurements to kind of get an idea about the function of what these genes do. Because as I said, only a fraction of all genes in the human genome currently are characterized and have attributed functions. Often they have one function, even if they're characterized, they have one function attributed to it, but many genes carry out many other functions as well that we don't yet know about. So we need unbiased and comprehensive approaches to characterize functionality, and we can do that now in the context of the patient cell, which I think is really the change. We don't have to resort to well-established laboratory cell lines, which anyway have an altered genomic composition, often than a normal cell has.
So the very rare diseases are kind of giving you a bit of a shortcut.
Sorry, I didn't understand.
They're giving you like a shortcut.
Yeah, Yeah, they provide, they're low-hanging fruits in a way because it's a one gene defect, and often the penetrance is very high. So if you have the mutation, you develop the disease phenotypes. So we don't deal with that complexity. Like if you're talking about complex diseases, it's usually a number of genetic factors together with a number of environmental influences, and you can arrive at the same ultimate disease state through many different avenues along that path. And so, you're dealing with heterogeneity that is really hard to dissect.
Oh!
How do you see how this kind of data will reflect on the health insurance companies and policies? Because I see the potential abuse of the patients here.
Yeah. I mean, there are all the issues associated kind of with taking advantage of it in a negative way that obviously needs to be prevented. But I won't kind of talk about that, but I will mention kind of for the healthcare system, I think it's actually going to be of interest to them to resort to this avenue because it's going to be an ultimate savings, right? Most of actually the deployment of money that goes into healthcare happens late in life. It happens to try to kind of maintain an individual that's already diseased in a sort of comfortable way so you can prolong life longer if it's kind of a detrimental thing or minimize symptoms. But at that stage, you have to take the medication continuously and it's expensive. However, if you can prevent it early, then you might intervene once and you change your course of action and then the individual kind of stays healthy. But the interesting kind of concept behind this and that needs to be solved is that the savings kind of here in this case, they come late, but the insurance company is going to have to pay for analysis early while the individual is still healthy. And so that has all these effects of, what about health insurance systems? In a case where the government provides insurance, then if they're paying for it throughout an individual's life, then they have an interest in that. If it's maybe a private health insurance that you're anyway going to change, they might not want to pay for it early because maybe at the time you would have developed the disease, you would be with some other company. And so there are all these things to take into account. But I think ultimately it's going to make healthcare cheaper and therefore will be supported by insurance companies.